As the brain signals the body, nerve impulses are sent down the axon, creating action potential. As the action potential reaches the synaptic knob, gated ion channels open and calcium ions are allowed to enter the cell. When the calcium ions, blue nerds, rush in, it causes synaptic vesicles filled with acetylcholine, green nerds, to fuse with the synaptic knob, releasing the neurotransmitter into the synaptic cleft via exocytosis. At the cellular level, let's look at the muscle group organization at a macroscopic level. Here at the thickest end, we have the fascicle, leading into a thinner myofiber, an even closer look of a myofibril. Now let's go in for a closer examination. Here we are at a closer look of the myofibril, which is organized into what is called a sarcomere. The sarcomere is organized or subdivided into different regions, which we will label now. The Z band, the red portion, thicker portion, the A band, the outer region, including all of this at the end, is the I band, and the central region we'll call the H band. The influx of sodium ions into the muscle fiber initiate an action potential on the outer membrane of the muscle fiber, also called the sarcolemma. Traveling along the sarcolemma into the T-tubules, the action potential signals the release of calcium from a region of the sarcoplasmic reticulum called the terminal cisterna. This is the region in which calcium has been sequestered by a protein called calsequestrin. Upon exiting the terminal cisterna, the calcium enters into the sarcoplasm of the muscle cell. After the calcium leaves the terminal cisterna of the sarcoplasmic reticulum, it attaches to the troponin complex. First, the calcium covers the thin filaments and binds to troponin, releasing tropomyosin from the actin active site. The exposed active sites attract high-energy myosin heads, which quickly bind, forming a cross-bridge. Myosin heads release ADP, converting to a low-energy state. This forces the thin filaments over the thick filaments, which is known as the power stroke. ATP then binds to the myosin head, releasing from the active site on the actin. ATP is then converted to ADP, which energizes the myosin head. Calcium is released from troponin and pumped back into the sarcoplasmic reticulum. At this point, tropomyosin shifts to once again cover the actin active site, finally allowing the muscle to relax. Acetylcholine, green nerds, binds with the acetylcholine receptors, green tic-tacs, on the motor end plate of the muscle. This causes sodium-potassium channels to open, allowing the influx of sodium into the muscle fiber and the escape of potassium into the synaptic cleft. Should we film the first scene? After acetylcholine produces its effect, it is removed from the synaptic cleft and degraded by a Pac-Man-looking enzyme called acetylcholine esterase.